let's see, what I want to talk to you about today is this work Christian described with the fruit flies about both the complexity of how, it's a very elaborate problem to try to think about how the bacteria are affecting the physiology of the fly, because the physiology of the fly is already very complex, and the bacteria are already very complex. But also there's the complexity angle, but also, how does the fly, actually specifically control individual members of the community to get what it wants out of the microbial community. And so what you're looking at here is the abdomen of a fruit fly. In cyan is it's a histone GFP. And so you can see individual nuclei. This is this kind of segment through the middle here is the gut and a midgut, and then you can see these kind of more in like orange kind of fibrils or trachea. And then there are ovaries up here, and lots of stuff going on. So it's a complex environment. What's cool about this image actually is this is a living fruit fly that actually we were able to peel back off of the microscope, and it walked away and lived for another month or so before passing on. So we can see a lot of this stuff. I won't really get into those details, but we can see a lot of this stuff in real time, which is giving us more and more insights into kind of the molecular details, the cellular details of how these systems are actually working. And so the microbiome to me is a really interesting problem just because it's really complex. There's a lot of stuff going on. It's also important for global health. There's microbes are involved in the global nutrient cycles, the carbon cycle, the nitrogen cycle. These affect global warming. They affect the ability of plants to extract nutrients from their soil and do their photosynthesis. They're also important for medical health, for animal health. They contribute to obesity, type 2 diabetes. I have these incidences here. I don't know what in Europe what the incidence of obesity or diabetes is, but if you get off a plane in the United States, it's immediately apparent that we have a problem in this country with these diseases. And the microbiome has been implicated. The composition of the microbiome, which species are there, are important for those diseases. So the way a lot of these, I was asked to give more of a detailed background. So the way that these phenotypes have been correlated between microbiome composition and human health are primarily from these survey studies where they take a population of people, they collect the microbial community, which is typically poop, and extract DNA, do some different sequencing techniques, do some bioinformatics, and then they can figure out what is the relative abundance of different species of bacteria in the gut, in that stool sample. And so different studies use different metrics. It could be which species are present, or it could be the abundances of those species. But the types of outcomes you get in these studies is that things like, here's the evidence that obesity is correlated with the composition of the microbiome. And so here's these, the blue dots are people who are not obese, the red dots are people who are obese. This is a principal component analysis. So it's really taking all the information into account of the different species that are present. The beta diversity was the input here. If you're more the ecological type, then this is PCA beta diversity, but really the red and the blue are separating. So we can see there's a correlation with the composition of the microbiome. And there was also causation of the microbiome. And so these are done with these GNOTOBIOTIC mouse studies where they can take the donor stool from a lean or an obese person, give that to a mouse that has no other bacteria in it. And then in this study by Peter Turnbow, when he was in Jeff Gordon's group, found that if the obese person's stool was fed to a mouse, those mice put on more body fat than if a lean person's stool was inoculated into the mouse and the mouse is eating mouse chow. But so there's causation established there. The difficulty here, though, is that the microbiome's got 1,000 species in it. Each person has a unique microbiome of roughly 1,000 species of bacteria. And so while we can say that there's a phenotype that's caused by the microbiome, we don't really know what is causing it. And despite 2 decades now of these sequencing surveys, we haven't identified causal species that are, the ones that are making a human or a mouse be obese or not. And so there's really a challenge in terms of the complexity of the community. There's so much variation and noise that we don't really know what it is that's actually in that mixture causing the problem. And the kind of the alternative hypothesis is that really it's not one species, that we would have found it with the depth of sequencing, the number of studies, that really it's the interactions between those species that are having the effect. And so that's one of the things I want to talk about today, is just how the interactions between the bacteria can actually be producing phenotypes. It's not just the individual bacteria, but really how is the whole different from the sum of its parts. And so a lot has been done on this. You're almost certainly familiar with genetic epistasis. And this is the idea that traits that we observe in animals and plants come from genes, and there are Mendelian traits where one gene is basically causing the trait, but there's also a lot of complex traits, and these complex traits have many different genes contributing to the one trait. So there's a pathway of gene A, gene B together produce phenotype C, and you can mutate A or B and see different consequences for the phenotype as you do different types of genetic manipulations. So these complex traits are more difficult to study, and so we've developed these frameworks to really just to say when a pathway is present or not, versus is it a Mendelian trait. There's a lot of different mathematical models for how to do that, and I'll also want to talk a little bit about that today. So the idea with the microbiome is that these bacteria are also carriers of genetic information. They're almost like a genetic locus, and that the interactions between them can also produce different phenotypes. And so you have the blue bacterium and the orange bacterium. They have a positive interaction between them, and but if you put in the pink bacterium, that has a negative impact on the blue one. It can have that can cause a negative impact on the orange one, and so you might see a trait that in an animal is caused by the orange one, but it can be indirectly affected by the presence of the pink one. So there's all these complex interactions, and they can have all these different effects, and so it makes it a really complex problem to study. And so we think about how does a microbiome affect an animal or a plant? You have to think about there's genetic interactions encoding these ecological traits for the bacteria. The bacteria have these ecological interactions within their host, and those interactions can be for instance pulling new nutrients in for the host, or synthesizing new nutrients. Could be negatively or positively affecting the immune system. They're affecting the physiology of the host and its fitness in its environment. So there's all these different levels of interactions that are really interesting and challenging to study. And so getting back to those challenges, if we want to try to understand them, we need to try to dissect these systems. And so one of the major challenges I alluded to earlier is that each person has a unique microbiome composed of roughly 1,000 species. And it's stable over time, like a fingerprint. So if I were to, sequence, if all of you listening in were to send me a stool sample, I could sequence that stool, and I would say that each person's got their unique microbiome signature. If we did a month later or a year later, I could do the same sequencing, and I could identify the people with pretty high level of accuracy just from the stool without knowing their identity based on what they had in the first data point. And so this gives this idea that this microbiome is unique to the individual and stable over time. So it's like a fingerprint. But what causes that fingerprint is really challenging to try to understand. And if the composition, if that fingerprint is contributing to your health, we want to figure out how we can really change the fingerprint. And so there's different aspects that contribute to that. Host genetics is one. There are indications that some host genotypes are, could be contributing to the composition of the microbiome in terms of how you get colonized by what. the diet feeding what's there. Maybe you just have a consistent diet that feeds certain bacteria and they persist in your gut for that reason. Your lifestyle, how often you run, how often you go to the bathroom could affect your microbiome composition, and your environmental factors like, are you just being exposed to different bacteria, different allergens, other things that could change your microbiome composition. So it's a very complex problem to try to dissect. And so one of the ways people are doing this is with model systems. That's what I work on. But this is a squid, which is maybe the simplest, most specific association between a bacterium and a host. And so, things like diet and environment, and there's a lot of random noise there, and it's really difficult to say, what is behavior versus what is actually a, governed by a molecular, ligand receptor interaction inside of a host, where there's real recognition of the bacterium by the host and the host by the bacterium, and changes in the physiology of them that drives this, coevolution between the two species. In a squid, it's very obvious that a squid is able to uptake specific bacteria. This is not a bobtail squid, but a bobtail squid is a squid that has a light organ, and many squid have a light organ where they uptake a phosphorescent bacterium from the water column. They find the specific strain that produces light. They put it in this evolved pouch that they have, this specialized pouch that holds it and keeps any other bacteria out. And it feeds it, it nurtures it over time. So it's able to produce light from these bacteria that it needs to, for instance, camouflage itself from predators in the moonlight at night. When a predator looks up and it sees glowing, it looks like the moon, it just kind of blends in with the moon. And they also apparently use them to hunt and communicate. But these light organs are, essential for the squid's fitness. And so there's this really tightly co-evolved relationship between the squid and the bacteria. We don't know that level of specificity in humans or really very few other animals, but there are some insect models that are really interesting, and fruit flies are one of them. And so my lab studies the fruit fly, and what's the reason to study fruit flies and not squid is that we're thinking about the gut microbiome, which is invaded continuously with new bacteria. And it's got a fluctuating nutrient environment. The diet, every time the animal eats, it gets a flush of nutrients, and there's a lot of random effects, and also a really high diversity of bacteria continuously coming in. So we need a model for that. And fruit flies are nice because, as opposed to the thousands of bacteria that we have, they've got much closer to a handful of species. And so different methods of isolating the bacteria produce different results. Different sequencing methods produce different results. But one of the things that my lab has done is take the wild flies and flush out anything that's transiently associated. And so that's gotten us down to between 3 and 8 species every time we do the experiment that are actually stably associated, really stuck inside of the fly. So we have this low diversity community. It's got lactobacillus, which are a human probiotic, a really well-studied human probiotic. And then these acetobacters, which are basically vinegar producers. But these are things that you find in food fermentations worldwide, probably because the fruit flies are bringing them and inoculating things. So I guess tell people, if you ever have had natural wine, they squeeze the juice and then they let the air bring in the bacteria or the yeast really that are fermenting the wine. But the thing that brings in those, it's not just the wind, it's the fruit flies. They come and they drink some of the juice, and then they contribute their microbiome to inoculate the culture that becomes the wine. So fruit flies, of course, also have these amazing genetic tools that things like squid and other insect models don't have. Probably the best studied developmental genetic model for animals. And then we've got sequenced genomes of all the bacteria, made them genetically tractable with plasmids. And then importantly, it's really easy to make a germ-free fruit fly. And then we can reassociate it with defined microbial community. So we have an incredible amount of control over the over the whole biological system when we're doing these experiments. And here's our 5 species. I'm going use these colors to keep track of them for most of the talk. And so I want to tell two stories and just actually see how far through I get. But the first one, I want to talk about how these, what Christian alluded to, how the interactions between the bacteria contribute to the host physiology. And the second aspect that I want to get at is how colonization works and how the interactions affect colonization. So to get to the first question, is the whole just the sum of its parts? So if we have, a 5-species microbiome in the fruit fly or 1,000 species microbiome in a human, how much is the whole really based on the individual contributions of the individual species members versus these species together have interactions, they use different sets of genes and different metabolisms with those different sets of genes and actually produce really different effects when they're together as a group. So the way we do this is work from Alison Gould and Vivian Zhang, who published this paper back in 2018. But the basic experimental setup is that we make each combination, we use a germ-free fly as a starting point, and then we either associate it with no bacteria or we associate it with each combination of bacteria. So we have 5 species, so we have 32 possible combinations, 2 to the 5th. There's each of the single strain associations, and then there are each of the pairwise associations, 3, 4, 5 species altogether. This is the way we set the experiment up. We inoculate the bacteria, we add the germ-free flies, and then we transfer them twice a week to fresh vials, and then we measure their phenotypes. And the phenotypes I'm going to talk about are bacterial composition, so like what do the fly do to the composition of the bacteria that we put in, as well as the lifespan of the flies and the fecundity of the flies. And so here's what some of the bacterial composition data looks like. If we look at just the red bacterium, this is the Lactobacillus plantarum strain, and we look at all these different replicas. There's a distribution of abundances of bacteria per fly, and so that's the kind of the normal variation that we see. If we look at all 32 combinations of the bacteria, we see a lot of variation, and we see that certain abundances change depending on the presence of other species. So, for instance, the yellow guy, there's more of it when it's on its own versus when this blue guy is present in certain cases. And so we can use that, we can ask the question, how does the abundance of red change when green is present? And if we do that, we can map the interactions between the bacteria contributing to their abundances. And so here is a food web where I'm using the data from 1 and 2 species diversity to calculate the interactions. If an interaction is positive or one species increases the abundance of the other species, then it has a black arrow. If it's negative, where it's decreasing the abundance of the other species, it has a red arrow. And so you can see that most of the interactions are positive, but there are also some negative interactions. If we look at the higher diversity, 3, 4, and 5 species data, we see that the interactions tend to be almost all negative, and so and that they change. So the interactions are depending on the context of other species. So you think about like a married couple may be intimate with one another when they're alone together, but when their kids are present, they have to be parents, and so they're disciplinarians, and they have all these rules that their kids hate, and their kids don't know them for the loving people that they might actually be. So the same kind of things would be happening in a microbiome, and we see these context-dependent interactions. So that's important because there's actually potentially genes that are expressed in these cases when microbiome the composition changes, and so gene expression programs could come on and off. And so not only does this tell us that there are interesting interactions in this community, but there are these context-dependent ones as well, which, at higher diversity, they're more negative, so they tend to stabilize the community. And so, this is potentially a way that we can actually do some, discovery of new biological gene clusters in these communities. And actually, I won't talk about that work, but that is something that we're doing in the lab, is trying to understand what are the genes that are coming on and off in these different contexts. In some cases, there's actually interesting, gene clusters that haven't been studied before. So the second part of this, of the first part of the talk, is just to talk about the interactions between the bacteria as they contribute to the host phenotype. So I keep showing this hypercube here, but there's this geometric representation of these of this experiment that we actually use to calculate interactions, and I will briefly touch on how we do that. But so what we see when we just measure the lifespan of the flies as a function of their composition is that there's overall a lot of variation in the single species associations, but there's a decrease in the variation and a decrease in the total lifespan as we increase the diversity in the associations. And actually is more clear on this graph. We see just the average lifespan decreases as a function of the number of bacterial species in the gut. And so that's puzzling because we hear all this that the microbiome is good for us and that lost diversity hurts us. And it's unclear, what this means for a fly versus a human in that context. But also, we know that certain species have certain effects and could potentially be changing things. And some of this could be based on the interactions between the bacteria. Some of this could be based on individual species traits. And so also had measured the fecundity of these flies at the same time. And so this also could be based on a decision by the fly to actually to allocate resources to fecundity rather than lifespan. Because what good is a lifespan out in the environment where I think people have measured the average lifespan of an adult fly out in the environment from some of these observational studies is like 3 to 5 days. And so what good is living 45 days out in the wild? So maybe they're just allocating their resources differently. And so when we plot the fecundity as a function of the lifespan.
There's a question in the chat if you want to check it,
I
maybe.
can't see the chat. Thank you for interrupting. Please.
So I can tell you, they're asking if the flies are adults or from L1. I think maybe it's regarding to larva.
Oh, I didn't explain that. Thank you so much. So these are all adult flies. So these are all, we rear them to 5 to 7 days post-eclosion, and then we associate them with the bacteria. So all of them have been reared identically up to the point when they're, it's about 5 days when they can actually get a microbiome in the first place. So they start, they're feeding more actively at about 3 days, 3 to 5 days, they're actually getting their microbiome. So we wait until they're all, this helps us have a really uniform starting population so that we're not having developmental effects that are actually driving this. So none of this is based on larval development or pupal development. Thank you for the question. Yeah. Do you have other questions?
Yeah, just a quick one. The relationship you showed earlier between bacteria, synergistic or antagonistic, are they inside the flies or on the food?
So both, actually. That's an important caveat of that that data that I was showing. So we haven't, so in that experiment, the flies are associated with the bacteria in the vials, and we're flipping them to fresh stock. We know that they are inside the fly, so those numbers are based on what's happening inside the fly, but we also know they're continuously eating bacteria from the food, and there's bacteria living on the food. So that's an important caveat. And we could talk more about the implications of that. But I'm going to talk later on about colonization, and that'll get more into the direct effects that we know are happening only inside the fly, because we've developed methods to isolate what's happening inside the fly so there's no feeding of bacteria from the food? Good question. Just getting back here, there's these associations between the so the lifespan of the fly is actually for a short lifespan fly, we actually see a higher level of fecundity. And actually, so when we were publishing this work, I actually had two very young children at the time, and I was getting very little sleep, and they were just destroying me. And this has made a lot of sense. The higher fecundity would lead to a shorter lifespan. So we wanted to ask the question whether the fecundity was actually causative of the loss of the decreased lifespan, or a consequence, an independent consequence of the bacteria in the microbiome. And so what we did, focusing in on a subset of these flies, and this is the same data now, I'm just showing, actually the error bars are too small to see on these lifespans, but you'll see them, I think, on some of the other ones. But these are error bars on the fecundity. But so we wanted to ask, so the experiment that we did was we started them with the bacteria. They do most of the reproduction in the first 3 weeks. They're basically done reproducing after 3 weeks, and then they live another up to 5 weeks, a total of 8 weeks. So we basically were asking, after 3 weeks, if we take away the bacteria with antibiotics, then what happens? Do they still have a short lifespan because they did all that reproduction? Or do they, are the bacteria what's killing them? And so then the loss of bacteria is going to extend their lifespan. And so what we saw is that in the vast majority of cases, the lifespan is extended by removing the bacteria. So it wasn't it wasn't their offspring or the reproduction that was killing them. It was actually the bacteria that were doing it. Interestingly, if we look at this subset, there's the green association, these acetobacters, and then this triple association between the Acetobacter orientalis and these two lactobacilli did not increase their lifespan when we removed the bacteria. So there's a memory, a physiological memory of having bacteria in these flies. This is a lifespan of 6 weeks, and we removed their bacteria at 3 weeks. So they really, they've been living just fine, and then all of a sudden they die as if they still have their microbiome. So this is curious, but what's also curious from a microbiome interaction standpoint is that the intermediate bacterial association of Acetobacter and this one Lactobacillus plantarum has a positive, does not have the memory, it breaks the memory. And so one of the things we're working on now is trying to figure out what is going on. Why does this one association have no memory of having a microbiome, whereas this more complex association, the simpler association, do have a memory? And so there's probably a physiological basis inside the fly, but it's based on this interaction. This is certainly a non-additive interaction with these phenotypes. So the interactions are important. And the kind of the conclusion of this work is that single species contributions have a limited amount of predictive power for what's happening in the complex associations. And based on time, I'm going to skip over this section, but this is just thinking about the different null hypotheses and how can we build models to really take the low diversity phenotypes or even the high diversity phenotypes and try to backtrack out what the interactions are that are causing that. And a lot of it depends on the hypotheses. I'm going to go briefly through this part here, which is a mathematical model that we developed in collaboration with Michael Josvig, Olga Ible, and Lisa Lamberti. And we've been working with a high-dimensional framework to try to understand these interactions, because one of the things that happens is that in these different types of models, they break down in higher dimensions. And so thinking about these fitness landscapes for epistasis, we've applied this type of thinking to the ecological frameworks inside the fly, really thinking of the bacterial genome as another genotype that we can add to the fly genotype. And so this is like an epistatic framework where we say, here's a discrete locus. It's a bacterial genome rather than a piece of the fly genome, but we can add it and remove it and then measure the phenotypic outputs based on that discrete genetic information. So I just want to give you the highlight of that work, which is that certain species have these huge effects on the interaction landscape between all the other bacterial species in terms of the effect that they have on the fly's lifespan. And getting back, I showed you before, it was a lactobacillus that was changing a lot of these interactions. And we see that as well when we do these, basically, any way we've done it, we see these really strong effects of the lactobacilli. They basically, all this is saying, and I won't explain it, but the lactobacilli change the interactions between the other bacteria and their effect on the fly's lifespan. And so these are major, like keystone species in terms of the, not really keystone species in the traditional definition, but keystone species in terms of being major players in the phenotypic output on the fly, which might be why lactobacilli are such a prolific antibiotic. So with that in mind, I want to switch over to thinking about colonization of the fly's gut, because how these compositional effects are determined by that are determining the physiology are determined by the ability to be colonized. And so Benjamin Obadiah was the first postdoc who worked with me at Berkeley, and he decided to take a very focused approach to understanding how an individual bacterium colonizes an individual fruit fly. An individual species of bacteria colonizes the fruit fly. And there had been this study, actually been a series of studies, and this is one of them, that it's shown that the fly microbiome was really non-specific, that basically whatever a fly has in it is just whatever it recently ate. And flies eat garbage, so of course they're going to have bacteria. There was this consistency in terms of which species, but that was thought to come from what was growing on the food, that they just kind of prefer to eat that, not that those bacteria actually has any kind of stable residence inside of the fly. And so there were a couple of caveats of this experiment that Benjamin addressed. One was that the developmental age of the flies, when they're 2 days old, they actually don't have a fully developed gut, and so these flies were being transferred to fresh food from the time that they had closed. So they're not actually eating right away, so being transferred away from the bacteria on their foods. They actually and they come out germ-free as well. So they basically never had a chance to be colonized, and then colonization really happens at about 4 days. And so Benjamin did all of his studies starting 5 to 7 days, which is based on this study as well as some other gut development studies. We decided to choose that time point. The other caveat, which I know that you guys think about a lot, is the ecosystem and the selection effects on the flies and the bacteria. So here you have two environments. One is the fly vial, and the other is the wild fruit or that a fly is eating, and where selection in the wild is actually happening. And so one of the caveats of this experiment was that they're using laboratory bacteria that were taken out of laboratory flies. And the environment for these flies, so there's about 10,000 to 100,000 bacteria per fly. And there's probably about 50 flies in a vial, and there's billions, maybe tens of billions or hundreds of billions of bacteria in a fly vial. And so selection is really being kind of dominated in the fly food on the vial. And so there's no selection for actually being maintained inside of a fly in this environment, because the fly does just eat a bunch of garbage and then get transferred to the next tube, taking its bacteria with it. Whereas in the wild, these are ants on here instead of flies. Sorry, I didn't get a good picture, but the fly is got to, this apple is going to dry up, or it's going to rot with something that's going to kill the bacteria. So the fly really has to tag along with the bacterium has to tag along with the fruit fly to get to that next piece of food, get to that right environment, that successional environment of the rotting fruit, where it's going to be able to proliferate and get into the next fly. So out in the wild is a very different environment, and we thought that maybe the wild bacteria would be important for understanding the fly biology. So, or for really studying the colonization. So Benjamin, taking in mind the fact that he wanted to think about the abundance of the bacteria, because the dose of bacteria could have a major effect, the exposure, the level of exposure could have an effect. He developed an assay where he could feed a defined dose of whatever genotype of bacteria we wanted to look at to a genetically defined fly, the lowest variation, all inbred fruit flies, zero variation from fly to fly as far as we could tell. Feed a very defined dose, a precise amount, and then he would passes them to fresh food every day for 3 days, and he'd surface sterilize them, crush them up, and count how many bacteria were inside. And he did this in 96 well plates, so we get high throughput. We've actually got another slightly higher throughput method now, where we do the same thing, but we can quantify things a lot faster. And this allows us really to study the variation in the traits, not just the mean, but also the variation. So what we see from these studies that there's actually stochastic colonization of the flies. And so here's each individual data point is an individual fly, and is the abundance of bacteria that it has in it 3 days after being colonized or being fed the initial dose, and then on the x-axis is the dose that it actually ate. And so some flies that, germ-free fly eats the most common bacterium found in flies, and it's at an abundance like 10 million bacteria, and some of these, have bacteria in them. The flies have bacteria, and some of them don't, even after eating that huge dose. And we made sure they ate the dose, or we excluded them from the study. So all these flies ate their dose. And so we see in the end point, 7 to 21 days later, is that there's this variation in the colonized flies in terms of the abundance of bacteria per fly, but there's also a bunch of flies that are germ-free. And so we can convert that to a colonization efficiency plot, where the y-axis is now the efficiency, the ratio of colonized to colonized plus uncolonized total flies. And so there's only a 50% colonization efficiency, but this purple strain came from humans, the human Lactobacillus plantarum. So we thought maybe there was some specificity for the host. And it turns out, the lab flies have a little bit higher colonization efficiency. And then when we look at the wild fly isolates of the same species of bacteria, any dose that we feed has 100% colonization efficiency. And so there's genetic differences between the bacteria that are contributing to the colonization. It's not just stochastic. So I say colonization works like a lottery. If this was a lottery, you would not want to play it, because this lottery is rigged. In the human isolate, you only have a maximum chance of 50% of winning the lottery, even when you buy all the lottery tickets. So there is both stochasticity as well as determinism in these bacterial strains for their colonization. And so one of the things I'm not talking about today is that we're getting into the genetics of what genes are actually causing this specificity for the host. And so formulating just a model of this behavior, we can think about the initial colonizers. They come in, and they're basically stochastically bumping into host tissue. But when they do bump in, they have a chance of forming a stable adhesion and embedding in this mucus layer. And then if they do so, then they have to balance. Then there's this stochastic growth problem of colonization from low initial abundance. The pioneer species comes in, and it has to balance the growth and the death rates, which are both also stochastic. And so it might go extinct, or it might form a stable colony, and then you have a stably colonized fly. And so we can formalize that in a mathematical model where we have the binding affinity up here. We have the likelihood, basically roll the dice odds down here. And then we can, we model both this model. It allows us to see both that there's a bottleneck in the population, that there's this dip in abundance in the early days that makes it more stochastic. And this is what cuts off the abundance, the maximum efficiency of colonization. And there's also a difference in just the per cell odds of colonizing when in this niche that we're calling it. And then taking this model up to kind of a total population structure level is work with Eric Jones and Gene Carlson and David Savak. We've been able to take this to the population level, and we see basically to see turnover in the population, that's going to tell us that how frequently these bacteria are shed and create new niche space to be colonized. And so we've done these experiments that I'll hopefully have a little bit of time to talk about, where we feed an initial dose of bacteria, and then we challenge them with a secondary colonization. We see what percentage of the flies become colonized by the secondary dose. And we see that different strains of bacteria have different phenotypes here, but we measure the mean once colonized, and we get the probability of colonization as a function of that. And that allows us to differentiate things that are strong colonizers that are actually forming a stable spatial structure in the niche versus ones that are maintaining themselves by a high growth rate, basically, in that same region. And so this makes a prediction that the Lactobacillus species has a really strong spatial structure. And so we've developed these methods of actually visualizing the spatial structure. So I'm showing you here is Lactobacillus, the strong colonizer, LPWF we're calling it, is labeled with mCherry in red. And DAPI stain of the fly nuclei is what you're mostly seeing of the gut is in blue. So here's a diagram of the fly gut, and here's an actual fly gut dissected out of a fly. This is the crop, which is a side appendage of the gut. It's a sac, storage sac. So when you shoo the flies away, they eat their food, and then they store it in their crop, and they fly away, and they go eat that. And save it for later, and then they pump it back into their gut later on. So in a regular fly, it's eating a lot of bacteria off the food. It's got bacteria all throughout its gut. There's maybe more in the posterior midgut than in the anterior midgut, but there's also a bunch in this foregut tissue, the duct going down to the crop, and this proventriculus tissue here. So it doesn't look like anything specific is going on when we look at that. However, when we flush the bacteria from the fly just by feeding it a bunch of germ-free food, we see only colonization up in this foregut section of the gut, and that's what we've identified as this colonization niche. Work by Rin Dodge has shown that the niche is only colonized heavily by these wildfly strains of bacteria. We see a little bit of colonization in the laboratory strain of the Lactobacillus, and almost no colonization in the human strain. So it's genetically determined by the bacteria. Here's just another view of that. And then here's a look at what the fly tissue looks like next to it. So the bacteria are like the black dots here, and these are yeast. They're autoclave yeast that are part of the fly's food that are just passing through. There's this cleared layer here that we don't. Evidence that I'm not showing you today is suggested it's mucosal. And then there's the fly cells that are here surrounding this. So you can really see, and there's this evidence of secretion here. So these bacteria don't look like the sausage shape that you associate with bacteria. That's because they're aligned, all coming out of the page at you. So you're just getting these circular cross sections of them. So they're all packed in this structure. Let's skip over this. So I guess I'm just about out of time. So what I'm going to do is wrap this up and skip ahead to show you that there is this spatial structure that the first colonizers get in, fill in these spaces, and that the secondary colonizers just get pushed right through the lumen, and they never have a chance to really touch and stick to these tissues where they're actually being accumulated. We can measure the half-life of the cells in the gut. So we have with this pulse chase scenario, pulse chase experimental design. So they have a half-life of about 2.5 days. which is very long in comparison to the 2-hour division time. So it looks like the dividing cells, most of them get pushed out into the lumen where they're shed, versus these resident cells are sticking around for quite a long time. And this doesn't happen with these laboratory strains and the human strains of bacteria. They actually get pushed out really quickly when we challenge them with a strong colonizer. Okay, I'm just going to skip over the rest of this, but there's spatial structure and there's multiple species. And what's I guess the interesting part is that the secondary species take advantage or are able to take advantage of the primary species in order to achieve colonization. They don't do a good job colonizing on their own, but when the first species is there, it allows them in. This is just some imaging showing that the host has a response. But so the key conclusion from the second part of the talk here is that flies have a symbiotic niche. They're maintaining this structure in their gut, and it allows them to keep control over this complex community of 5 species of bacteria by really controlling the first species. So the specific pioneer species, Lactobacillus, induces these changes in the fly tissue that facilitates these other symbionts to colonize. And so this provides us a model in the fly to understand how the host can use specificity for certain strains in order to manage complex associations with other strains that actually have these big benefits on its fecundity. So the Acetobacter is the secondary one that I was talking about. That earlier in the talk, I was telling you that it really contributes to the fecundity. And so this is just my kind of conclusion slides, are that the interactions between the bacteria determine the colonization as well as the... At what point did you lose me?
Well, when you started with your conclusions about the interactions.
Okay, cool. So that wasn't too far back. So I'm back. So basically, can you hear me again?
Yes.
Okay, so I was very quickly go through the basic idea is that there's all these different interactions between species, but that the fly can and they could produce different combinations of bacteria based on those interactions, but that the fly is able to exert control over which species are having the initial colonization, and that allows it to control the kind of the pathway, the trajectory of colonization to get to the desired microbiome composition that's going to favor its fitness. And so what the secondary strain that was coming in was this Acetobacter that contributes to fecundity. And so the fly is basically able to prepare itself to receive that strain by first getting the lactobacillus. And so I'm just going to skip through here, but so the idea is that the fly is able to control this complex association. I want to thank Ren Dodge did a bunch of that work. He's a technician in the lab. Halong Ju is a grad student in the lab who's doing the fly genetics. So I didn't show any of that, but that's upcoming. And then that was the main contributors. And then I have these collaborators who've worked with for a lot of the math modeling. So with that, I'd love to take your questions.